A 2003 study at the University of Toledo found that 30 seconds of horizontal eye movements, left to right, right to left, measurably improved episodic memory retrieval in adults. 30 seconds. Not surgery, not medication, not a device. If you are over 55, your corpus callosum, the bridge connecting the left and right halves of your brain, is shrinking at an accelerating rate right now. And what you're about to learn is how a simple, repeatable eye movement may be one of the most direct ways to force both hemispheres to communicate again. Most people believe their brain works as one unified organ, one solid mass doing one coordinated job. That belief is wrong. Your brain is split, physically split, down the middle. You have a left hemisphere and a right hemisphere, and they perform very different tasks. Your left side handles language, logic, sequential processing. Your right side handles spatial awareness, emotional context, pattern recognition. And the only way these two halves talk to each other, the only physical bridge between them, is a thick band of approximately 200 million nerve fibers called the corpus callosum. Here is what almost nobody over 50 has been told. That bridge is deteriorating. A 2025 study published in Communications Biology using data from the Human Connectome Project in aging, 718 adults ages 36 to 100, found that callosal tract density remains relatively stable until around age 60. Then it drops sharply. The networks that decline fastest are the ones responsible for attention, executive function, and motor coordination. The salience and somatomotor networks showed the steepest losses, explaining over 43% of age-related structural variance. So when you walk into a room and forget why you're there, that's not just getting old. That's your left hemisphere encoding the intention and your right hemisphere failing to retrieve the context. The bridge between them is getting thinner. But the real question is this: can you force that bridge to work harder, even as it shrinks? The answer involves something most neurologists never discuss with their patients, and it starts with your eyes. Your eyes are not just input devices, they are motor organs. And when they move, particularly when they move horizontally, left to right and back again, they activate a cascade of neural circuitry that reaches far beyond vision. Think of it this way. Imagine your brain as a house with two rooms separated by a hallway. The left room stores your words, your plans, your logical sequences. The right room stores your memories of faces, your sense of where your body is in space, your emotional impressions of the past. The hallway is your corpus callosum. Now imagine that hallway gets narrower every year after 60. The rooms still work, but the messages between them get slower, weaker, more garbled. That is what age-related callosal atrophy does to your thinking. When you perform a horizontal saccade, a deliberate left-right eye movement, you are doing something neurologically specific. You are activating the frontal eye fields, located in the prefrontal cortex, on both sides of your brain simultaneously. The frontal eye fields are the command centers for voluntary eye movements. When you move your eyes to the left, your right frontal eye field fires. When you move them to the right, your left frontal eye field fires. A rapid left-right pattern forces alternating activation of both hemispheres in rapid succession. Dr. Stephen Christman and Dr. Ruth Proper at the University of Toledo published a series of studies between 2003 and 2008 demonstrating that just 30 seconds of bilateral horizontal saccades improved episodic memory, the type of memory that lets you recall specific events from your life. In one experiment, strongly right-handed participants, who typically have smaller corpus callosum volume and less natural interhemispheric connectivity, showed the most dramatic improvement. Their recall accuracy after bilateral saccades nearly matched that of mixed-handed individuals, who naturally have larger callosal connections. Think about what that means. In 30 seconds, an eye movement temporarily compensated for a structural disadvantage in the brain. Now, researchers still debate the exact mechanism. Some believe it is a direct increase in cross-hemispheric coherence. A 2011 study at Leiden University, led by Samara and colleagues, used EEG to test this hypothesis. They found that bilateral saccades improved recall of emotional words, but interestingly, did not produce measurable increases in interhemispheric EEG coherence. The behavioral improvement was real. The proposed neural explanation didn't fully hold up. So what is actually happening? The research is still evolving on this, but a competing theory from Dr. Keith Lyle at the University of Louisville suggests that saccades don't just link the hemispheres. They activate attentional control networks within each hemisphere. The frontal eye fields connect directly to the intraparietal sulcus, a region critical for episodic memory retrieval. Saccadic eye movements may pre-activate this region, priming your brain for better recall. Either way, the behavioral data is consistent. Your eyes move, your memory improves. But this is only half the story. What happens at the structural level, inside your aging white matter, is what makes this relevant to everyone over 55. The corpus callosum is not a uniform cable. It is a mosaic. Different sections connect different brain networks, and they age at different rates. This is where the 2025 Human Connectome Project study changes everything we thought we knew. Researchers at multiple institutions, Washington University, University of Minnesota, Massachusetts General Hospital, and UCLA, tracked callosal fiber density across 7 major brain networks. What they found was striking. The fibers connecting your salience and ventral attention networks, the circuits that decide what deserves your attention and what to ignore, showed the fastest decline after age 60, with cubic trajectories explaining 43% of variance. Your somatomotor network fibers, the ones coordinating movement, declined almost as steeply at 42%. Your default mode network, the system active during daydreaming, memory consolidation, and self-referential thinking, showed a 31% variance explanation. But here is the finding that should concern you most. The relationship between callosal density and cognitive performance is not constant across age. In younger adults, there was almost no correlation between how thick your corpus callosum was and how well you performed on memory and attention tests. Your brain had enough reserve, enough backup routes, enough plasticity to compensate. After 60, that changes dramatically. The correlation between callosal density and cognitive performance became strong and significant. In older adults, higher tract density in the somatomotor network predicted better verbal memory, with a correlation shift of 0.338. In the salience network, it predicted faster processing speed, with a shift of 0.320. If you've ever woken up and felt like your thinking was foggy for the first hour, like your brain was booting up slowly, like you couldn't find the right word even though you knew it was there, this is what's happening inside your white matter. The bridge is thinning, and your brain no longer has the reserves to compensate. So when researchers like Christman and Proper showed that bilateral saccades could temporarily enhance episodic memory, they were demonstrating something more important than a parlor trick. They were showing that the brain's cross-hemispheric circuitry can be externally stimulated. Not with drugs, not with implants, with eye movements. If this is clicking for you, if you're starting to see why this matters more than most of what you've heard about brain health, subscribe now. Because what comes next is the part that most people, including most doctors, have never heard. And it changes how you think about your daily routine. A 2013 study by Parker and Dagnall, published in Frontiers in Human Neuroscience, tested whether bilateral saccades specifically improved episodic autobiographical memory versus semantic memory. 69 participants performed either horizontal saccades, vertical saccades, or no eye movements before a memory fluency task. Horizontal saccades improved the retrieval of specific personal events from both childhood and adolescence, but had zero effect on the recall of factual autobiographical information like names of teachers or friends. Zero effect on general semantic knowledge like naming animals or vegetables. This is a critical distinction. The saccades are not making you smarter. They are not boosting general knowledge. They are selectively enhancing the type of memory that lets you re-experience your own life. The kind of memory that deteriorates fastest in early dementia. The kind that makes you feel like yourself. But none of this matters if you don't understand what happens to your brain's connectivity during sleep and why that 15-second eye movement may need to happen at a very specific time of day. What almost nobody talks about is the relationship between your corpus callosum and what your brain does while you sleep. During deep sleep, specifically during slow wave sleep, which dominates the first half of your night, your brain generates large, synchronized electrical waves that sweep across both hemispheres. These waves are not random, they are coordinated, and the structure that coordinates them is the corpus callosum. A study published in the Journal of Neuroscience by Avanuti and colleagues examined split brain patients, individuals whose corpus callosum had been surgically severed. In these patients, slow waves generated in one hemisphere failed to propagate to the other side. The cross-hemispheric sweep was broken. The callosum was essential for it. Why does this matter to you? Because slow wave propagation is not just an electrical curiosity. It is one of the primary mechanisms by which your brain consolidates memories during sleep. Your hippocampus, the short-term memory vault, replays the day's events during slow-wave sleep, and that replay needs to reach cortical areas in both hemispheres to be properly stored as long-term memory. If your callosal connectivity is weakened, that replay signal gets degraded, crossing from one side to the other. This is where the science gets genuinely disturbing. The same networks that decline most sharply after 60, the salience, somatomotor, and default mode networks, are also the networks most involved in memory consolidation and motor learning during sleep. So as your callosum thins, you lose connectivity during the day and you lose consolidation capacity at night. It is a double hit. Depending on who you ask, this may explain why sleep quality and memory decline track so closely in aging populations. Not because sleep itself is broken, but because the hardware that moves information between hemispheres during sleep is physically eroding. Now here's the counterintuitive part. Some researchers believe that horizontal saccadic eye movements generate a neural signature that partially mimics what happens during REM sleep. During REM, the dreaming phase, your eyes naturally make rapid horizontal movements. This has been documented since Azarinsky and Kleitman's original 1953 discovery at the University of Chicago. Interhemispheric EEG coherence increases during REM, and that increase has been specifically linked to the presence of horizontal eye movements during REM phases, according to research by Dion in 1986. Stop and consider that for a moment. Your brain naturally uses horizontal eye movements during REM sleep to enhance cross-hemispheric communication. And when you deliberately perform those same movements while awake, your episodic memory improves. You are not inventing a new brain function. You are manually triggering one that already exists. You are pressing a button that your brain presses every night, except that as you age, your brain presses it less effectively because the callosal fibers carrying the signal are thinner. Historians of neuroscience still argue whether Francine Shapiro's original 1989 discovery of EMDR, eye movement desensitization and reprocessing, was a clinical insight or a lucky accident. She noticed her own distressing thoughts diminishing as her eyes spontaneously moved back and forth during a walk in the park. That observation led to a therapy now endorsed by the American Psychological Association and the World Health Organization for PTSD treatment. The therapeutic mechanism may be different from the memory enhancement mechanism, but the underlying principle is the same. Horizontal eye movements change how your brain processes and integrates information across hemispheres. So the question becomes, if your brain has this built-in mechanism, and if it deteriorates with age, how do you use deliberate eye movements to support it? Here is a specific, evidence-based protocol built on the research we've covered. This is not speculative. Every step traces back to published findings. Step 1. Perform 30 seconds of bilateral horizontal saccades every morning, within 30 minutes of waking. Sit upright. Hold your head still. Focus on a point to your far left, then shift to a point at your far right. Move your eyes, not your head, back and forth at a pace of roughly 2 full cycles per second. That is approximately the same rate used in the Christman studies. One position change every 500 milliseconds. Do this for 30 uninterrupted seconds. Why morning? Because your cortisol is naturally peaking in the first 45 minutes after waking, and cortisol enhances the encoding of new memories. By priming your interhemispheric circuitry while cortisol is elevated, you are creating optimal conditions for the day's first round of memory formation. Step 2. Repeat the 30-second saccade protocol immediately before any cognitively demanding task, before reading something important, before a medical appointment where you need to remember instructions, before studying new material. The Parker and Dagnall research showed that the memory enhancement effect is time-limited, likely lasting between 4 and 9 minutes, based on related findings from Shobe and colleagues in 2009. So timing matters. Do the saccades immediately before the task, not 20 minutes before. Step 3. Add a 15-second variation before bed. This time, slow the pace. Move your eyes left to right at approximately 1 cycle per second, half the speed of the morning protocol. Some clinicians using EMDR adjacent techniques suggest that slower bilateral stimulation helps activate parasympathetic tone, potentially supporting the transition into deeper sleep stages where callosal dependent slow wave propagation is most critical. This application is more speculative than the morning protocol. Some scientists dispute whether such brief pre-sleep stimulation produces measurable effects, but the physiological logic is sound and the cost is zero. Step 4. Combine saccades with a single leg balance test twice per week. week. Stand on one leg with your eyes open and perform slow horizontal saccades for 15 seconds. This forces your vestibular system, your visual system, and your motor cortex to coordinate simultaneously across hemispheres. It is an integration exercise. Dr. Jean-François Delven at the University of Leeds published a 2025 review in Neuroscience and Biobehavioral Reviews, documenting that callosal integrity directly affects balance and gait function in older adults. Training balance while stimulating bilateral eye movements is targeting both systems at once. Step 5. Track your results with a simple self-test. Once per week, read a short news article. Then wait 10 minutes. Then try to write down every specific detail you remember. Names, numbers, locations. Do this for 4 weeks with the protocol and 4 weeks without. You will see the difference. If even one of these steps was something you had never heard before, hit like. It tells the algorithm to push this information to more people who actually need it. This is not entertainment. This is applied neuroscience. Let's step back for a moment. Your brain was not designed to last 80 years in a body that sits still. It was designed to move, to scan, to track prey across a savanna, to watch for threats in peripheral vision. Every horizontal eye movement you made for hundreds of thousands of years reinforced the connection between your two hemispheres. Your ancestors didn't need a protocol. They needed to survive. And survival meant constant bilateral visual engagement with the world. Now you sit. You stare at a fixed screen. Your eyes barely move for hours. And the bridge between your hemispheres, the structure that holds your cognition together, quietly loses density year after year, with no symptoms until the deficits are measurable. But it does not have to go that way. The data from the Human Connectome Project shows that callosal integrity predicts cognitive performance more powerfully in older adults than in younger ones. That means the people who need this information the most are the people who benefit from it the most. Your brain at 65 is more dependent on that bridge than your brain was at 40. Protecting it is not optional. It is structural maintenance. I want to hear from you. Drop a comment below. Can you do the 30-second saccade test right now? Left, right, 2 cycles per second for 30 full seconds. How does it feel? Did anything surprise you? I'd bet most of you have never deliberately moved your eyes this way, and that itself tells you something about what your brain has been missing. Your hemispheres don't stop working, they stop talking. That is the line to remember, because everything in this video comes down to one physical reality. The connection between the two halves of your brain is a living structure that responds to use and decays with neglect. 30 seconds of deliberate eye movement is not a cure, it is a conversation between two halves of a brain that forgot how to speak to each other. Share this with someone who needs it, a parent, a partner, a friend over 55 who has been blaming their memory on age instead of on architecture. And if you haven't subscribed yet, do it now. What I've been researching for the next video takes this even further, into territory that connects your eye movements to your autonomic nervous system in ways the science is only beginning to map. Your brain is listening. Move your eyes. Start the conversation.